Today we're diving into epigenetics, which is the control of that, right? So epigenetics is the study of how gene expression is regulated without changing the underlying DNA sequence. Epi means control. It explains how the same genetic code or change, right? Control or change. But it explains how the same genetic code can produce very different outcomes, right? Depending on the signals the body receives. These signals come from lifestyle, all the things we've been talking about, environmental food, right? Stress. So your internal environment, your external environment, your stress levels, your emotional state, and even exposure to toxins and supportive inputs like nutrients and herbs, right? So at the core of epigenetics is the idea that genes are not fixed in their activity. They can be turned on, they can be turned off, they can be adjusted in intensity. This regulation determines how cells function, how the immune system behaves, and how inflammation is controlled. One of the primary mechanisms of epigenetics in the control is DNA methylation, and that's what we talked about last week, specifically MTHFR and the inability or ability to methylate. So when methylation is insufficient or improperly placed, genes that should be quiet, such as those driving inflammation, may become overactive. Proper methylation is essential for controlling your immune responses. It's essential for controlling detoxification. And it's essential for controlling cellular repair. Another mechanism used is histone modification. So DNA is wrapped around proteins called histones. And this is the way it is wrapped is determines whether a gene is accessible or hidden. So when DNA is tightly wrapped, the gene is less likely to be expressed.